All right, so let's continue with where we were. We were at the bilaminar embryo, right? So our bilaminar embryo we created with the epiblast cell on the dorsal side and the hypoblast on the ventral side. And what we're going to do is go through the process of gastrulation to create our trilaminar embryo now. In this case, I'm going to kind of stop making the trophoblasts. It's a little bit easier for me to just kind of focus on what's going to happen inside of what was the embryoblast. So gastrulation is going to create 3 layers. It's going to create 1, 2, 3 layers. Just remember, pause, like, what are those 3 layers? Endoderm, mesoderm, and ectoderm. Okay. So we're going to do the first things first is we're going to build our epiblast layer, right? So there's our epiblast layer, and that created my amniotic cavity. And then I'm going to build my hypoblast, but in this case, I'm going to draw it a little bit far away to give myself enough room for what's going to happen in gastrulation. So there's my hypoblast, which creates my yolk sac. And I'm going to transition and change colors just so you guys understand what's happening. Okay, so the first thing that's going to happen is on the dorsal side of our bilaminar embryo, we're going to start to create an indent. And that indent is going to go from the tail region to the head region. So it's going to go caudal to cranial. So caudal to cranial. Probably cranial. I put caudal right here. That is the primitive streak. Sometimes it's referred to as the primitive groove. The streak is a little bit less of an indent. The groove is a little bit bigger. So essentially what's going to happen is you're going to start to have a little depression here on the dorsal side, and our epiblast cells are going to kind of thicken right on the lateral edges right there. And as those epiblast cells thicken on the lateral edge, they actually have room to go other places. There's a little bit of space between the epiblast and the hypoblast. Those cells are going to start to migrate internally to find room. And when they migrate internally, they're going to first displace the hypoblast layer, and they're going to first create the endoderm. So you can see how the endoderm and the yolk sac will still kind of be continuous because those layers are still there. You just want to think about the hypoblast just helps create the part of the yolk sac. And then as those epiblast cells continue to migrate internally, they're also going to give rise to more cells in this middle layer. And those cells in the middle layer end up becoming the mesoderm. So as our epiblast cells continue to come in internally to make the mesoderm cells start to cluster at the midline. And when they cluster at the midline, they form the very first structure that we see in our embryo. Remember, that's the notochord. Okay. Let me change my position of my little part right there. So all that layer is my mesoderm. And then essentially by default, the epiblast cells are no longer epiblast cells. So I'm going to make them a darker blue color. Those epiblast cells will still have that little indent, and they become the ectoderm. So that's gastrulation and how we create our 3 layers of our trilaminar embryo. Yes, you still have your trophoblast out here. If you're worried about it, there's the trophoblast layer going all around, okay? So the next part here, in this next part, there's kind of an open space because what you're going to do is go through several processes to make your trilaminar embryo more complicated. Remember, we're going to do neurulation. which is going to create the dorsal hollow nerve tube. Mesoderm is going to become more complex. And we're going to roll up the gut tube. So remember, we're going to do all these things simultaneously. So I'm going to kind of draw them in sort of a simultaneous way, but I'm going to start with neurulation because neurulation kind of should happen first. So we're going to take that thick blue layer that was our ectoderm. We're going to build that first. So there's my ectoderm. I'm still going to have my primitive streak, my primitive groove a little bit along the dorsal aspect of my embryo. And what's going to happen is I'm actually going to start to differentiate the ectoderm into two types. So right where the primitive streak is, we're going to make a purple layer, and that purple layer is going to become neural ectoderm. And that neural ectoderm is going to start to get thicker at the lateral edges of the primitive streak. And that's going to eventually cause us to start to have this U-shaped structure, which is the neural groove. And the higher areas are the neural folds. Okay. At the top of the neural folds, we're going to make a little bit of an orange structure. This is where we form neural crest. If you have a hard time with neural crest, just think about, like, if you're thinking about sound waves have a crest and a trough. The crest is actually the upper part of the sound wave. So that projection that goes upwards. So we're going to create this nice neural groove and these folds that come up. And then the other blue part becomes our epidermal ectoderm. Okay, so at the same time our mesoderm has started forming, we should already see we have a nice block of notochord right there. And we're going to leave some space. Oh, hopefully I can draw a straight line. Let me draw a little bit farther away, like that, to give myself some room. So that's my mesoderm layer, and then I'm just going to make a squiggly endoderm layer because the endoderm's not going to do too much. And I'm going to come back to that picture in a minute. We're just going to continue with neurulation. So what's going to happen is your neural ectoderm is going to start to form that neural groove even more. So instead of having just a U shape, you start to have what looks like a circle. And the neural crest cells are going to come together. And at that point, you're going to see that the edges of the the epidermal ectoderm are going to start to come together. And as they do that, we're going to see that we create a hollow tube along the back, and that hollow tube is the dorsal hollow nerve tube. Then you want to create the notochord, anterior or ventral to that, and create a line for your mesoderm. And then just create some endoderm. Perfect. So now you've created your epidermal ectoderm, which is going to create your skin. Your dorsal hollow nerve tube, which starts in the middle of your embryo and then zips towards the head and the tail at the same time. And that's going to create your brain and your spinal cord. Your mesoderm now is also going to start to get more complicated at this time. So we're going to go back to our upper picture and see what happens to our mesoderm. So in this case, like if we go back to our picture here, the mesoderm is just, you know, like filling the space. It doesn't have any distinct features. As you go through neurulation, you're going to start to see that the mesoderm kind of starts to take a little bit of shape, like this. And eventually, as your dorsal hollow nerve tube completely rolls up, you're gonna start to see that there's individual discrete blocks of mesoderm that form lateral to the notochord. Dorsal hollow nerve tube actually will be a little bit bigger right here, but it's easier to draw it this way, okay? These are called the somites. Somites are going to differentiate into 3 layers, so I kind of draw it like this, and we're going to have a dermatome, a sclerotome, and a myotome. So when you look on your chicken embryo, you should see a nice thicker area on the somite. That's the dermatome. And then I want you to come in here and make your own notes about what the D is. So you can like kind of maybe do that right here. D equals, M equals, S equals, and then think about what it makes. What does dermatome make? What does myotome make? What does sclerotome make? Are they segmental or are they transsegmental? What are they derived from? From the somite. What's the somite derived from? Mesoderm. And then just lateral to that, I usually kind of draw a little bit of a rectangle or square, and that becomes the intermediate mesoderm. Remember, the intermediate mesoderm is segmental, so that means every section I'm not necessarily going to see that. And then on the lateralmost edge, this block, or this, I shouldn't say block, it's a sheet. This sheet of tissue ends up forming the lateral plate mesoderm. Remember, lateral plate mesoderm is going to run the entire length of the body. And as we progress through this, the lateral plate mesoderm is going to get more complicated, and it's actually going to split into 2 parts. So there is going to be this part that comes up underneath the ectoderm, and a part that actually goes on top of the endoderm. So lateral plate mesoderm splits into 2. This portion that goes underneath the ectoderm is called the somatic mesoderm. The part that goes above the ectoderm is called splanchnic mesoderm. And then what happens is you create a fluid-filled cavity between them called the coelom. Just like that. The last part is to figure out what happens to our endoderm. The endoderm stuff, how this rolls up into a tube is actually much more complicated to draw, so I actually have a picture instead. What this is going to be the fact that we're rolling up to create our body plan. This is called transverse folding or lateral folding. So if you want to think about it, you can think about gut tube creation. Okay. So if we kind of go backwards a little bit, I know this might be counterintuitive, but if we go backwards and we go back to our bilaminar embryo, we kind of think about how things are being formed. In our sagittal view, what's going to happen is our epiblast cells are going to start to move internally from the tail to the head. And that's going to help create our mesoderm from our tail to our head, which is why the notochord gets formed from the tail to the head. And our dorsal hollow nerve tube is going to roll up on the dorsal surface, but it's going to start in the middle of our embryo. So we're going to start creating our tube right about here, and then our tube is going to continue towards the head and towards the tail simultaneously. So this is why you create the notochord first, and then the dorsal hollow nerve tube. And as you sort of finish creating the dorsal hollow nerve tube, the gut tube is going to start to roll up. So how are we going to roll up the gut tube? We want to just come through on this picture and label as many things as we can. So this is amniotic cavity. If you don't know that, at least try to find this is the ectoderm. If you want to, epidermal ectoderm. And then hopefully you know what that is. Hopefully you know what that is. If you don't know what this is, let me go over that on the other picture really quick. The only thing I forgot is right about here and right about here, you'll have little circles in your splanchnic mesoderm, and that's going to create the dorsal aorta. I can't draw right there because my little thing is here. Let me just gonna remove my endoderm really quick and put dorsal aorta. So the dorsal aorta are paired first, and then they're going to come together to form one dorsal aorta. So there's my endoderm again. So I want you to label those yourself. Can you label that? Can you label that? The only thing that this picture is not good at is where is the somatic and where is the splanchnic mesoderm. So what we're going to see is you should kind of draw in a red portion here, and then that red portion goes just underneath, just deep to the ectoderm. That is the somatic mesoderm. The splanchnic mesoderm is going to be closer to the endoderm, or where the gut tube is going to be formed. So this one I'm going to help you draw and label. This is somatic mesoderm. This is splanchnic mesoderm. So as the lateral edges of our ectoderm come together and fold up, this is going to create the endoderm becoming a tube. And that tube is the gut tube. Okay. What happens to the yolk sac? The yolk sac kind of creates a sac that's still outside of the body temporarily, just at the midgut area. And eventually the gut tube and the yolk sac are not really combined together because the yolk sac goes away. Which I spelled splanchnic wrong here. Make sure you guys spell it correctly. Okay. Don't worry so much about what happens to the yolk sac. We'll learn about that in 2240. So the whole point here is you roll up into a tube. And when you roll up into a tube, so the lateral folding creates the basic body plan. So I'm going to do this on another video, so we kind of chop it up into smaller condensed pieces. How to draw your basic embryo and how to apply that to adult cross sections.